not sure why they're all women. Men get these two, of goiters, and a goiter is an enlarged thyroid gland. That is the result of an absence of negative feedback, as we'll see today, and a result of the trophic effect of the TSH. And I'll give you two examples of why negative feedback is important. It's important in all axes where you have a complete axis, but I put illustrations in both of these. Are there any questions from last time? No? Alright, so last time we talked about the hypothalamo-pituitary adrenal axis. We talked about CRH, which works through a G protein mechanism, a G alpha stimulatory. We're going to learn about another kind of G protein today. And it essentially can both change the activity of the cell, phosphorylation events. We didn't go into detail. But also it can change gene expression through its interaction with CREB. Right? So remember, G protein activates adenylate cyclase, which activates pKa, which could phosphorylate CREB and induce transcription. So it actually changes gene expression for the ACTH in the corticotroph. Right? The ACTH circulates, and the ACTH functions in the adrenal cortex, in the zona fasciculata, to promote an increase in cortisol. And we talked about several ways that that happens. When ACTH binds, sorry, somebody say something? When ACTH binds, the first thing it does, or the fastest thing, it does all of these things, is it increases the release, make sure that's all right. It increases the release of cortisol that's already stored in the cell, in the liposome. And this is the only case of a steroid hormone that's actually stored in the cell. I told you that was analogous to releasing the burgers that were sitting under the heat lamp. So CRH, the store manager, recognizes there's some stress somewhere, tells ACTH, the shift manager, to tell the adrenal gland to put out more cortisol. The fastest thing that happens is the release of stored cortisol. The second fastest thing that happens is that you get the enzymes to produce more cortisol. And that's done by phosphorylating those enzymes. Those enzymes include the desmolase, or the 2022 lyase, that takes cholesterol and converts it into pregnenolone. Those enzymes include the delta-5 delta-4 isomerase / 3-beta-HSD. It's physically the same protein. The same enzyme does both of those functions. If I were you and I go on an exam and you had to write it, I would write both of those down just to acknowledge that you know both of those things are happening, right? It's a 3-beta-HSD because it's changing from a hydroxyl group at the 3 position to a ketone. And it's a delta-5 delta-4 isomerase because it's flipping the double bond. Same enzyme is working down here to go from 17-hydroxypregnenolone the 17-hydroxyprogesterone. The enzyme is acting here as a 21-hydroxylase. Again, you should be able to tell me without knowing. As the 21 position, you've added a hydroxyl group. In this case, it's the 11th position, you've added a hydroxyl group. It's 11-hydroxylase. So not first, all of these things are happening. It's just that the fastest thing that happens is to get the burgers that are already made out the window. The stored cortisol is released. The second fastest thing is you make the enzymes work faster, work harder. So you phosphorylate the enzymes. The next fastest thing, and the rate limiting step, is you get that cholesterol and that cholesterol that's in the outer mitochondrial membrane into the inner mitochondrial membrane. I told you that's analogous to getting the frozen burgers out the freezer. So those enzymes all can work faster than getting the cholesterol out of the mitochondria. That's the rate limiting step. Oh, and I was reminded of something just now. The next fastest thing you do is you upregulate all the enzymes. That's the equivalent of hiring more employees. So ACTH, and you could already tell me how, because it works through cyclic AMP, adenylate cyclase, cyclic AMP, PKA, and CREB. The genes that are regulated by ACTH are the genes for these enzymes. So there are genes for the enzymes, there are no genes for the steroids. Don't say that on my exam. And you'd be surprised, like 3 or 4, I just had my nephew with me this weekend, I call them knuckleheads. 3 or 4 of you knuckleheads will still say something about the gene for a steroid. I'm sorry, I shouldn't have said that, wasn't I? Don't say it. That's my point. So the next fastest thing is to upregulate the transcription and translation of those enzymes, hiring employees. The last thing, under extreme stress or absence of negative feedback, is you have a trophic effect. You build a bigger crystal burden. You build a bigger adrenal gland. A couple people ask me questions. You do that by increasing the number of cells, primarily. I'm going to give you a counterexample today. But primarily, you increase the number of cells, and that happens because ACTH stimulates IGF-2. IGF-2, am I going too fast? Say something. This is your class. I already got tenure. That may happen to me, but if you don't keep up, you're going to be in trouble. ACTH stimulates the release of IGF-2. IGF-2 works in a paracrine, maybe even an autocrine function, to increase the size of the zona fasciculata, that portion of the adrenal cortex that responds to ACTH. Now, so, what's your question? Wait, what's your name first?
Victor.
Victor, what's up, Victor?
You said increase the size of what?
Of the adrenal cortex, the zona fasciculata specifically. And somebody asked me yesterday, I think it was Ulysses, IGF-2 does that through its own receptor. So IGF-2 is produced, it binds its own receptor, and stimulates the proliferation of cells at that part of the adrenal cortex. You had a question. What's your name?
Daniel, you answered it.
Oh, did I answer it?
Yeah.
Okay. I'm like clairvoyant like this. Yeah. Wait, are you high?
Yeah.
All right. I'm Tyrone. I'm just happy I remember somebody's name. This is a hard thing for me.
I cannot see why it's increased transcription of the genes downstream. How does it actually phosphorylate those proteins?
Because remember, the G protein activates adenylate cyclase, which generates cyclic AMP. Cyclic AMP activates PKA, protein kinase A, and protein kinase A phosphorylates a whole lot of things. It probably doesn't do it directly. If you want to know the whole pathway, take your first class. But it phosphorylates a whole cascade of things, and one of the end products that it phosphorylates are the enzymes responsible for making cortisol, and that activates them, that makes them more active. But probably not directly. I mean, that's the whole point of it. One hormone binds its receptor, it activates a number of G proteins, which activate a number of adenylate cyclases, which generate a number of cyclic AMPs, which activate a number of PK. So the whole signal is magnified. And that's why hormones can work at such low concentrations. The last, well, we're going to skip one part and save it for Thursday. It's suffice it to say now that the cortisol circulates with a binding protein called CBG, corticoid binding protein. So glucocorticoids like cortisol, and this is actually going to be important later. It circulates bound to one of these amphiphatic or amphipatic, whatever the word is. You guys learn the right words so you don't be like me. I just can't remember, I'm too old. So it circulates bound to one of these binding proteins. And some studies suggest that there's actually proteases at the site where cortisol is needed that cleaves the binding protein so that you concentrate cortisol. That's the only case of where I know that might be true. So some studies suggest that there are actually proteases that recognize and bind the CBG. So if somebody punches you, you have a bruise and you need cortisol there, when the cortisol comes by, the protease cleaves the protein and now you concentrate cortisol at that site of injury. We're good? Yeah? What's your name? That's what I was going to call you though. What?
Can you just see that part again?
So cortisol circulates bound to this corticoid binding globulin, CBG. And at some people, there's some studies that suggest at sites where it's needed, there's a protease that cleaves the CBG, so the cortisol becomes free and concentrated at the site. So there may be proteases at the site of an injury, for example, that help concentrate the cortisol, that helps cortisol find its target. It'd be like my old friend and student Melinda, remember the one who had the key and didn't know where her apartment lived? Once I figured out where her apartment was, I would like stop and kick her out of the car, so that would help her figure it out. So it's kind of like that. Analogies, they work for me. Cortisol works, well, cortisol has some fast actions as a glucocorticoid. It may act through cell surface receptor, but the primary way that cortisol works is through a binding protein that regulates transcription, the glucocorticoid receptor. And let's leave it at that, because we'll spend most of tomorrow, or Thursday, talking about how steroids work. So let's just leave that one right there for now. In terms of degradation, cortisol does something unique. What's the enzyme that does that? An exam coming up in what? 3 more lectures. Got big something. What enzyme does what I just did? Did I do it right? 11, yeah, good. 11-HSD. So cortisol is degraded by 11-beta-HSD. You don't have to remember the betas and alphas. Into cortisone. And this, I would tell people in Alzheimer's, this is one of the reasons I'm so fascinated with steroids and steroid biochemistry. The receptor can tell the difference between one proton of that big old molecule, which has a molecular weight of like what, 3-something or something like that. It can tell that that one proton is missing. And cortisone is no longer active. So when you hear people in the store, and when you buy something called hydrocortisone, that's the same thing as cortisol. So hydrocortisone is cortisone with the H at the 11th position. Hydrocortisone and cortisol are the same thing. But that's how it's degraded, but it's reversible. So the cortisol, you'll see later in the course, can circulate. 2 interesting things I just told you. One is the tissues can help cortisol find its target by cleaving the CBG. The second one is places that don't want cortisol, like the kidney. I'll tell you why later. You hate it when I say that, don't you? I'll tell you why later. Keep you in suspense. No spoilers here, buddy. Gotta show up. If cortisol goes in the kidney, that enzyme's waiting there to deactivate it. So if you get to a place that's not a target, it deactivates it, but it's reversible. It doesn't degrade it to the point you have to get rid of it. It can go somewhere else and turn back into cortisol. But tissues that don't want it, like the kidney, that's very important, deactivate it. That's how it's degraded. But it's reversible. We're good? Any questions? What's up, Maggie?
Can you repeat all of them?
All of that started from, hey, welcome back, or all of that started? All of that coming back to the first day? What, about the cortisol?
Yeah.
So 2 potentially important things, or interesting anyway. One is cortisol has a CBG. And at tissues, at tissues that are the target for cortisol, in some cases there's a protease that would cleave the CBG. And now the cortisol becomes free and concentrated at the site. So it's kind of helping it find the target. On the other hand, in places where you don't want cortisol, okay, I'll tell you, I'll give you a spoiler now. Cortisol can bind the aldosterone receptor, and that would be bad. We'll see. So at places where you don't want the cortisol, like in the kidney, they use this 11-beta-HSD to turn it into cortisone which can't bind the receptor. Which is crazy when you see the structure of aldosterone. They can't tell the difference between aldosterone and cortisol, but they can tell that one proton is missing from that 11th position. I don't know, that just fascinates me. Y'all don't look too excited about that. So you got to go to sleep. We're good? Yes?
So you also said that cortisol has a receptor on the cell wall, cell surface?
There is some evidence that cortisol, and actually several steroids, have these receptors on the cell surface that when bound to will elicit fast reactions. So cortisol is a glucocorticoid. Corticoid because it's, well, oid because it's a steroid. Corticoid because it's from the cortex of the adrenal gland. And gluco because it regulates glucose. So it mobilizes glucose. So there's some fast action of cortisol is almost like what you see for epinephrine. And you'll see later progesterone, estrogen also have some cell surface receptors. But the classic way that steroids work, as we'll talk about on Thursday, is through these transcription factors.
So it's usually the slow action rather than the fast action?
Most of what cortisol is known to do. It's a ligand for a transcription factor. Okay? And we'll have an entire lecture, or most of the lecture on that on Thursday. Yeah, one more. And then we get to go. Did I miss something? Cortisone? What's missing? Oh, yeah. Thank you. So the HPG is a little more complicated. Actually, the HPG is a lot more complicated than we're going to talk about today. I'm going to give you a simplified version of the HPG. There's GnRH, which I already gave you as abbreviation. You got the handouts out, right? Stands for gonadotropin releasing hormone. It controls two gonadotropins that come from two different cell types. The luteotrophs make one called LH, luteinizing hormone. It's named after what it does in the female. It induces ovulation and leaves behind something called the corpus luteum, which means yellow body. So after you, in fact, the doctor can look at your ovaries and tell you how many times you've ovulated based on the scars left behind from what this hormone does. But some little scope thing. FSH comes from the folliculotropes. They're in males and females. And it comes from the folliculotropes. Those are two different kinds of gonadotrophs. Let's stop there for a second. You got to learn a new mechanism now. GnRH binds to the gonadotrophs. It's a decapeptide. It's transcribed and translated as a prohormone, so a bigger protein, but it's a decapeptide, 10 amino acids. It binds and it activates a G protein, but now there's a different kind of G protein. Remember I told you about the G alpha? Oh, sorry, am I going too fast? Y'all say something then. Because I was at the perfect pace for me. So remember I told you there's a G alpha S, the G alpha stimulatory protein. That's the one we talked about most. There's a G alpha I, the G alpha inhibitory protein. That's what GHIH or somatostatin works through. This was called a G alpha Q. G alpha Q is a GTPase, just like the other ones. The beta gamma subunit inhibited the G alpha. But when GnRH binds its receptor, again, it's a transmembrane, integral membrane receptor, it activates this G protein, this G alpha Q. Have you guys had this stuff before? Raise it. Everybody close your eyes. No, seriously, close your eyes because people don't like to raise their hand if people look. How many people have had all the G protein classes before? Okay, so only a few. All right. So this G alpha Q, what it does is it activates something called phospholipase C. And phospholipase C cleaves from the cell membrane a lipid called PIP2. I'm probably misspelling this. Phosphatidylinositol phosphate is going to cleave that into two components. One is diacylglycerol, which is a lipid. And IP3. Inositol phosphate. So PIP2, which is actually a lipid in the membrane, it's actually on the inner side of the lipid bilayer, is going to be cleaved into diacylglycerol and IP3. Diacylglycerol stays. Am I going too fast?
Yeah.
Why don't y'all say something then? Say, hey, stop. I need to give you guys like a safe word for making me quiet. What's your name again? I forgot.
Anne.
Anne.
Yeah. So, I'm sorry, this is like way back.
Don't be sorry.
Why?
This is your class.
Why did you?
I've had it 20 times. What's the question?
Why did you erase G beta?
Oh, because the G beta and the G gamma move away.
Oh,
When the
okay.
GnRH, that's the thing I can do on the board. You can't do it on the paper.
Yeah.
So when the GnRH binds, the G beta G gamma subunit inhibits the G alpha
Okay.
in all those G proteins. But when the hormone binds and contacts it, it sends the G beta and gamma away, and then that allows the G alpha to bind the GTP and do its thing.
Okay. What hormone makes the G beta G gamma go away?
In this case, it's GnRH.
Okay.
But anytime a hormone binds that has a G protein coupled receptor, what it does is it makes the beta gamma subunit, which are inhibiting it, move away, and then the G alpha subunit then becomes active. Okay? And in this case, it's a G alpha Q. Actually, I think it's called Q/11. I don't know why. And I have no idea what the Q stands for. The S and the I know what they stand for. The Q, I don't know. So here's what happens next. The diacyl, and I sort of drew this here. Because the diacylglycerol, let's do that different. The diacylglycerol sort of stays in the membrane. That's the lipid part. This part, which is a carbohydrate, it goes, watch this. It goes to the, you know, the books call it the sarcoplasmic reticulum, but I was always taught that was only in muscle. Is that what you guys are taught? Yeah, so I don't know why it's called that. But it's a part of the endoplasmic reticulum that stores calcium. The IP3 has a receptor on the endoplasmic reticulum that causes calcium to come out. The diacylglycerol activates a kinase called PKC, protein kinase C. And I think it's called C because it's regulated by calcium. Wait, Ulysses, you had a question at the end of class. What the answer? Oh, yeah. Okay. I read your mind. You didn't have to ask it. You're like, right? Okay. I think the C stands for calcium, but it's protein kinase C. In fact, there's not a protein kinase B, is there? I never heard of it. So yeah, I think the C must stand for calcium, not that it's the third one in the group. So the PKC can then also open some calcium channels from the outside in addition to phosphorylating other things. And the PKC is a peripheral membrane protein. Remember what that is? That was, sorry, what was the young woman who helped me out? What was her name? Pearl? Where's Pearl? Oh, that was you? Okay, right? Remember, Pearl couldn't leave the wall. It's a peripheral membrane protein. So PKC can slide around and phosphorylate things. Just like the G proteins. They're kind of bound. They're not in the membrane, they're peripheral. The diacylglycerol is in the membrane. Okay, now, this is the last part. All this calcium then can activate they bind the calmodulin. I think 4 calciums bind each calmodulin or something like that, and activate cytosolic kinases that are calcium dependent. So lots of stuff is getting phosphorylated, changing the cell physiology. We're good? The important thing is, so lots of things get phosphorylated. Take Gary Firestone's class if you want to memorize all those pathways. This is as far as we're going to go for today, and for the course probably. The other thing that's important is that calcium is important for membrane, what's it called? For vesicles to bind to the cell membrane. And there used to be a wonderful video that showed how it worked, but they took it offline, and I can't find another one. So the other important thing is that all this calcium into the cell, into these gonadotropes, ultimately, if this were a luteotrope, ultimately lead to vesicle binding to the membrane and the release of the stored gonadotropins. We're good? I'll drink some tea while y'all catch up. What's up,
Is
everybody?
it then what PKC did?
PKC does a lot of stuff. This is the extent that you have to know. PKC is a cytosolic, PKC is a peripheral protein kinase. So it's phosphorylating things that are associated with the cell membrane, including calcium channels and allowing more calcium to come in. The calcium coming in through the endoplasmic reticulum and extracellular activates calcium-dependent protein kinases that can go all over the cell and phosphorylate stuff. That influx of calcium is also responsible for the binding and release of vesicles that are storing protein. In this case, LH. We're in a luteotrope. Well, we might not get to the cover now. Yeah, what's up? What's your name? What? Kareem, what's up? So calmodulin is a protein that binds calcium. I think 4 calciums bind one molecule, and together calcium calmodulin activates these calcium-dependent protein kinases. So the calcium doesn't bind to those directly. Through calmodulin, you activate these bad things. And
Is the process different to
no, same thing. The difference is, so the question is the process different for FSH? Here's the difference right here. I'm going to tell you the difference right now. The difference is LH and FSH are both big glycoproteins. They have this, see, I stole more crayon and Crayola. This is why both my kids majored in biology instead of art. I stole all their art supplies. So they both have the same alpha subunit. So the folliculotropes and the luteotrophs are transcribing and translating this thing, but they have a different beta subunit. So LH might look like this, and FSH, so you can stick them together, kind of Crayolas. FSH might look like this, but the alpha subunits are the same. So the exact same thing is, well, sort of. In terms of what GnRH does, the exact same thing is happening in the luteotroph and the follicular troph, except one makes LH and one makes FSH.
So we could easily replace LH with FSH.
Absolutely.
Okay.
Works the same. And it's more complicated. We got GnRH and inhibin and activin. We'll talk about all that later. This is all you have to know for now. Especially for females, it's a little more complicated. Yeah.
Do you need calcium to come from both of those places in order to activate the protein kinase? Or if one part of that pathway is not working well, will the protein kinase still be activated?
What's your name?
Nausea.
Nausea. Oh, I've asked you before. So the question is about the relative contributions of extracellular versus intracellular calcium. I don't know the answer to that. So there has to be enough calcium to make those things happen. How much comes from inside or outside? I don't know. I don't know anybody knows. But both things are happening. What? Wait.
I'm pretty sure calcium is regulated so that it's much higher always inside the cell than outside the cell. Because it'll mess up memory potential.
Yes, but she's asking which, in terms of these mechanisms, which contributes more, the intracellular release or the extracellular
I'm
release?
pretty sure it's the intracellular.
She says it's the intracellular.
I'm almost positive.
She's almost positive. Yes. Yeah.
Does GnRH only determine the LH and FSH getting out of the cell? Does it not have a part in making LH?
See, that's an interesting question. So there's a literature that says, actually, that regardless of this, so they do these pulse chase experiments, and there's a literature that says that most of what gets released is new protein. And some people have even questioned why you even store protein. So certainly some old protein gets released, but also there has to be some mechanism by which you continue to transcribe and translate more. And there are many, many pathways where that might happen. This is as far as we're going to go here. Yeah, one more, and then we got to make some steroids. You guys just don't want me to join more steroids. That's why I asked you questions. What's your name?
Tanya.
Tanya, what's your question?
So what did you say about whenever there's a G protein and something binds to something? I don't know.
You can't give me more than that. so I think you're talking about the question that Maggie, or I forget who, Ann asked maybe. So there's always a beta gamma subunit that inhibits the G protein and prevents it from binding GTP and being active. But when the receptor that's coupled to that G protein is bound, the beta gamma subunit are kicked out, and then the alpha subunit can bind the GTP. What the G protein does depends on whether or not it's G alpha stimulatory, G alpha inhibitory, or G alpha Q. And I don't know what the Q stands for. Okay. So the LH and FSH circulate. And a couple things can happen depending on if you're male or female, and depending on if you're female, what time of the month it is. If you have already ovulated, the LH stimulates you to produce progesterone from what's called the corpus luteum. And you've already learned that. LH works, how do you, oh, I'm going to go into this. LH works as a G protein coupled receptor. It works through a G alpha stimulatory. Just like ACTH does. And you already know the path. When LH binds in the corpus luteum after you've ovulated, you take your cholesterol, you create pregnenolone, and you go to, I didn't write it up there, and you make progesterone, and you stop there. Why do you stop there? Why do you stop there, do you think? Why don't you go ahead and make cortisol? You know why? Because all those employees that go on to make cortisol don't show up for work in the corporate luteum. All you got is the guy who puts the meat on the burger. Nothing else happens. That doesn't make any sense to y'all, did it? In my head, it sounded good. The enzymes, the rest of those enzymes aren't there. The corpus luteum only has the desmolase and the delta 5 delta 4 isomerase / 3 beta HSD. Doesn't have those other enzymes. So LH works the same way. It binds, activates the G protein, which activates the NLH cyclase, which generates cyclic-AMP, which activates PKA. What's the only difference? What can the shift manager in the adrenal cortex do that the shift manager in the corpus luteum can't do? I already told you today. If you have to make more progesterone, what option are you missing? The corpus luteum is like in and out. What? Storage. Yeah, no other steroidogenic tissue stores steroid. In and out, right? They make all theirs fresh. So the corpus luteum is more like in and out. They have no burgers sitting under the heat lamp. Everything has to be made fresh. So when the LH binds, it can phosphorylate enzymes, so you make more progesterone. It does that. It can phosphorylate star, so that you move more cholesterol. It does that. It can upregulate, and you should be able to tell me how. It can upregulate the two little enzymes that it needs to upregulate. And it makes your corpus luteum grow, and it maintains it. And it's by increasing the number of cells that LH does. And then if you get pregnant, there's another hormone that does it. And there's negative feedback. So if you make too much progesterone, there's negative feedback. As you'll see later in the course, this is how birth control pill works. You give progesterone, high levels of progesterone, it inhibits GnRH, LH, and FSH through negative feedback, or as a progestin. So that you don't grow any follicles. You don't get pregnant because you don't have any follicles developing. You generate an artificial negative feedback so that you aren't making LH and FSH and GnRH. You can do it with estrogen. You can also do it with testosterone, but then you grow a beard, and I guess you wouldn't have to worry about it. Oh, sorry, that's not. But any of those steroids would negatively feedback. Also remember, negative feedback is an action for that hormone. If you're a guy, that's sort of true. If you're a guy, LH stimulates the production of testosterone. Testosterone has negative feedback effects, but mostly because it's converted to estrogen in the brain and pituitary. You're going to see later in the course, testosterone doesn't do a whole lot. This is why, if you take anabolic steroids, if you're taking artificial steroids, because you want big muscles, so you're an athlete like hippies do when I was in high school, this is why your testicles shrink and you become infertile, because of the negative feedback. Not permanently. What's up?
Your testosterone and progesterone negative feedbacks, LHS, FSH, does it mean that FSH also makes testosterone, or?
No, but it's feeding back on both the GnRH and on both the gonadotropins. But FSH is not involved in making testosterone. I'll tell you what FSH does in a second. Let's make some testosterone first. In the testes, in the testicles, testicle just means testes that are outside of the body. Only mammals, except cetaceans and a few others. You know what cetaceans are? Whales, dolphins, yeah. I have to check sometimes. Here's what happens in the testicle. You make this weak androgen called dehydroepiandrosterone. What enzyme does that? Kind of a trick question, kind of not. I'll tell you what enzyme does that in a minute. You can also take the 17-hydroxyprogesterone and make another weak androgen irreversibly. Called androstenedione. Androstene, andro because it's an androgen, stene because it has this double bond, dione because it has two ketones. And this can be reversed. Oh, and these can go in between, right? Same enzyme, you know that. And this is our old friend testosterone. Testosterone is one of these portmanteaus. Did I tell you guys this? That means testicular hormone, testosterone. It's two words stuck together. What enzyme makes testosterone? You notice, you guys are supposed to study this weekend. You had 3 days. All day yesterday. It was too hot to go outside, so I'm sure you can study your enzymes. What position is this? 17 beta HSD. The enzyme that does this is what? What type of it? 17 what? 1720 lyase. Now here's the tricky part. The 1720 lyase is physically the exact same protein as the 17 hydroxylase. It's the exact same enzyme. So you have the potential, and you'll see some conditions where you'll do this, you have the potential to make androgens in your adrenal cortex. But it just so happens is that this 21 hydroxylase has a really low energy of activation. Is that the right word I'm using? So instead of going to here, the 21 hydroxylase pulls the 17 hydroxyprogesterone to make these glucocorticoids. I'll say that again. Was that too fast? Yeah. Don't say something. So this 1720 lyase is physically the same protein as the 17 hydroxylase. It's an enzyme with two different functions. In the adrenal cortex, you with me? You make these 17 hydroxylated progestins. You with me? This is like church. I gotta say something. I said, you with me? Yes. Give me anything. Then the 21 hydroxylase has such a low energy of activation. That's the right word I'm using, right? It goes easy. That it pulls this stuff over here instead of going to there. You don't get to these androgens because the 21 hydroxylase pulls it away from the enzyme. If you're a female, I'll give you to draw that over again. If you're a female, if you're a sexually mature post-pubescent female, premenopausal, in the first 2 weeks of your menstrual cycle, you do the following. The LH stimulates testosterone production just like in the male, right? So the granulosa cells of your ovaries, the thecal cells in your ovaries, the interstitial thecal cells do exactly what the Leydig cells do in the male. Why does it sound like I had an accent? M-A-L-E. We good? If you're a female, first 2 weeks of your menstrual cycle, the thecal cells of your ovary do this. Exactly what the Leydig cells do in the testes. But then FSH in your granulosa cells, which are inside your follicle, The FSH activates the G-alpha stimulatory protein, which activates PKA, which turns on CREB. And CREB together with another transcription factor called SF1 stands for steroidogenic factor 1, and it's involved in all steroidogenesis. Interacts with CREB. Steroidogenic factor 1. In fact, it's in the same family as the steroid receptors, but it doesn't have a ligand. We thought we discovered a ligand for it once and published it. Turns out that's probably not true, so don't believe everything you read. And here's what happens in your... What's up? Your name is Dan.
Yeah.
What's up, Dan?
When do you use that one for? SF1?
SF1 is a transcription factor that, along with the phosphorylated CREB, which we already talked about, upregulates the gene for an enzyme called aromatase.